I'm Songhee Ra, and today's lesson is about the cell cycle. Within the video, we will cover binary fission, the prokaryotic cell division, then sexual versus asexual reproduction, mitosis, meiosis, and finally the difference between mitosis and meiosis, mainly focusing on the difference between the steps and chromosomes. Prokaryotic cell division or binary fission is the process of cell division within a prokaryotic cell. Within the first step, copies move towards the opposite ends of the cell as the chromosomes duplicate. Then the cell elongates and chromosome duplication is complete. Now, what is the difference between asexual and sexual reproduction? In sexual reproduction, it involves gametes or sex cells. This also means that sexual reproduction happens through meiosis, not mitosis. Since it is the creation of gametes, they are haploid, meaning they contain only one set of chromosomes. This is because when an offspring is created, they get one haploid from each parent, making the resulting organism's cells diploid. Unlike asexual, sexual also has slower reproduction rate, but they also have the advantage of being able to adapt faster, since all offsprings are unique, while in asexual, they are all identical. Additionally, in asexual, it involves the replication of body cells, or also known as somatic cells. They are diploid and are reproduced through mitosis. Mitosis starts at interphase. This is where the cell performs normal life functions. Then it goes to early prophase, where the duplication of DNA and centrioles happen. The centrioles begin to move into position, and by the time they are at opposite poles, spindle fibers attach to the chromosomes, lining up on the metaphase plate. In metaphase, chromosomes align at the metaphase plate. Centrioles begin to pull the chromosomes apart, and by anaphase, the duplicated chromosomes split from the originals. Each chromatid goes towards one centriole. In a telophase, nuclear material surround the chromosomes and the cytokinesis happens, which is when the cytoplasm divides into two. I didn't previously mention this before, but interphase also includes its own steps. In mitosis, the duplicated chromosomes divide and evenly distribute to make two daughter nuclei. In the step of G0 in interphase, this is where differentiated cells or cells with not enough resources are at resting or non-dividing. These types of cells can be stem cells or cells like nerves. If a cell has enough resources and the right conditions, G1 or the first gap happens. Here, the cell grows in size and prepares for the DNA to replicate. Between all these steps, there are checkpoints and growth factor proteins to help determine if the cell can continue through the cycle or differentiate. Then the next step after G1 is S, the synthesis of a DNA. Here is when DNA is replicated. Single chromosomes become two sister chromatids. Then in G2, the second gap, the cells grow even more to complete preparation for cell division. If a cell didn't grow to its required size, it would lack many essential organelles, causing more problems. This concludes interphase. However, since the remaining steps like prophase, metaphase, and anaphase, and telophase are very important to meiosis too, we will review those next. These are also known as PMAT, and prometaphase isn't always considered a step sometimes and can be included as just prophase. In prophase, chromatid tightly coils and folds. mitotic spindle fibers form and move away from each other. In prometaphase, spindle microtubules attach to the kinetochores and it moves the chromosomes towards the center of the cell. In metaphase, the mitotic spindle is formed and the chromosomes line up at the equatorial plate, which is the mitotic, the metaphase plate. And in anaphase, sister chromatids are separated. Finally, in a telophase, the cell elongates, the daughter nuclei and nuclear envelopes are formed. When the two cells are separated, the chromatin fibers uncoil. However, there is a difference between plant and animal mitosis. First one is within the step of telophase. In plants, instead of a cleavage furrow in animals that shadow indentations in a cell surface for animal cells, there is a cell plate with vessels fuse to form a membranous disc in plant cells. Additionally, plants lack centrioles, which make up spindle fibers. We have been talking about mitosis for a while, but what about meiosis? It's just as exciting and quite frankly pretty similar. Meiosis does not have interphase. Instead, it has two PMAT cycles. There is meiosis 1, then meiosis 2. In early prophase 1, chromosomes and centrioles duplicate. Centrioles begin to move and chromosomes line up in homologous pairs. Spindle fibers begin to form, and by metaphase 1, chromosomes are lined up at the metaphase plate with microtubules attached to one homologous each. Anaphase 1 contains microtubules that pull and separate the pairs of chromosomes, and in telophase 1, cell membranes constructs, and the chromosomes are surrounded by nuclear material. In the end of meiosis 1, the results are two haploid cells, and the chromosomes are still duplicated. This is why we have meiosis 2. It is essentially the same as mitosis, but starts with a haploid cell. In prophase 2, spindle fibers form and move chromosomes towards the middle of the cell. Metaphase 2 chromosomes are aligned at metaphase plate. In anaphase 2, centromere of this sister chromatid separates and moves towards the opposite poles. And by telophase 2, nuclei are formed and at the cell poles, which allows for cytokinesis to happen right afterwards. At the end of meiosis 2, there are 4 haploid cells or gametes with no duplicated chromosomes. We have now successfully covered both mitosis and meiosis. Here are the differences listed. Mitosis is asexual, the division of somatic cells. 2 daughter cells are reproduced with 2N, 2 sets of chromosomes, 46 total chromosomes, only goes through PMAT once, but has interphase. And in meiosis is sexual, the division of gametes. 4 daughter cells are produced, has N or 1 set of chromosomes, 23 total chromosomes, and 2 sets of PMAT cycles with no interphase. And that is all for today's video, so see you next.